everyone. I hope that you're doing good in this cold morning here in West Lafayette. We want to introduce Dr. Adam Sparks, who is an alumni from the Department of Agronomy here in Purdue University. And later he did a PhD in Kansas State University with Dr. Karen Garrett. And like his expertise are botanical epidemiologists and and ecologists specializing in simulation modeling and GIS. He also is a co-founder of Open Plant Pathology, along with Dr. Emerson Del Ponte, and that is like a community for plant pathology, plant disease epidemiology, pathogen population biology. So he will be talking about this topic eventually, and we just want to appreciate like your time, and thank you so much for being awake in this time and give us this opportunity to interact and learn about your research. Thank you so much.
Thanks for that introduction, Mariella. Yeah, I'm sorry about the odd time, but it is 9 p.m. here in Perth. So it's a little bit odd for me, but I'm actually.
Oh, sorry, Dr. Sparks, I think I just muted you accidentally.
That's all right, I've got it. And what I was saying wasn't that important anyway, I don't think. Yeah, I've had a bit of an interesting career since I left Purdue. I left Purdue in 2000 when I graduated from agronomy, and ended up in Nebraska for a bit, and then drifted my way down to Manhattan and did a PhD at Kansas State. From there, I ended up going to the Philippines, where I did a postdoc initially at Erie in the plant disease management epidemiology group, which I later ended up leading that group when Serge Savary left. And then 2016, moved to Australia to the University of Southern Queensland. I was an associate professor of summer crops there. Hi, Levente, I see my former center director has joined the Zoom here from Queensland. So I spent 5 years in Queensland, University of Southern Queensland, leading summer crops pathology and doing epidemiology work, some of which I'll talk about here. And then in January this year, I moved across from Queensland to Perth. So basically a New York to LA sort of move, and joined the state government in Western Australia as a bioeconomic modeler. So I can no longer claim the title plant pathologist, but I do still have my fingers in lots of plant pathology projects. But I have a little bit of what I'm working on in this presentation as well, just to give you an idea of some of the things I do that aren't necessarily plant pathology. So with that, I'm going to share my screen and get started. Maybe, sorry, I'm trying to get my presentation synced up here so I can see. There we go. All right. So I'm going to talk about using maps, models, and open science and plant pathology today. And I want to start off with a caveat. George Box was a statistician, and I guess you could say he was a famous statistician, if there was such a thing, who said essentially all models are wrong, but some are useful. And I always like to open any modeling presentation with this, just so that people are reminded that models are a gross simplification of reality. You cannot capture all the processes, but if you can distill it down to something useful oftentimes, and when we derive use from the models, then we see the greatest benefit. So I'm going to start off with a broad, high-level application of using models to look at how does climate change affect rice disease in Tanzania. So this was a policy guiding exercise funded by GIZ, the German Development Corporation, while I was working at Erie. So this was the part of the Mikordea project that I did with folks from Africa Rice. And the project was mitigating the impact of climate change on rice disease resistance in East Africa. And the idea was that Africa Rice and others were interested to know what diseases are going to be affecting rice as we go into the future. And so if we're setting breeding priorities, what do we need to really be focusing on? So to do that, we set up a project where we did simulation modeling, and we took advantage of the situation, and we actually linked two models together. And so Serge Savary and Letitia Williquet have written a whole bunch of really useful models. One of them is rice pest, which is a production situation driven crop growth model. And it's got a really simple crop growth driver in it that then allows you to feed in solar radiation and temperature and precipitation. And it gives you attainable yield. And then the model accounts for yield loss due to injuries from pests and diseases, which then gives you the model's estimated attainable yield and therefore the amount of yield lost due to pests and injuries. So we start with a production situation, which is your fertilization and your irrigation, et cetera. So in this case, we were looking at, I think we used an upland rice simulation that was not really overly fertilized, but it had a little bit of fertilizer on it, and it was rain fed, it wasn't irrigated, sort of thing. So this is what we mean by production situation. The model then uses losses due to pest injuries and gives you your actual yield. So this is what it's capable of simulating. You can see here there's a whole slew of diseases, bacterial blight and brown spot, leaf blast, neck blast, sheath blight, sheath rot. It also includes weeds because it's production situation driven. A lot of work went into this from Serge and Letitia to determine that oftentimes there was a linkage between what weeds occurred and how heavy the infestation was in the production situation. And also the occurrence of insects, so the brown plant hopper and the defoliators and whatnot. And it had relatively simple data requirements. And like I said, the output was attainable and actual yield. So if you're familiar with crop growth models and the DeWitt models, this would be nothing new. And if you're not familiar with it, don't worry. I'm going to show you what this actually turns into. The second model that we used was was EpiRice. EpiRice is a disease severity model that predicts disease severity on a 1 square meter area, and in this case for a 120-day growing period. And we used weather data from, well, this is using weather data from NASA Power, which is freely available daily weather data on a global global grid. And this map, if you look at the left side at about 20 degrees latitude, shows you India, and a little bit down and to the right there is Sri Lanka. And then if you go across, you'll go through Bangladesh and Myanmar and down through Thailand. Indonesia goes across the bottom. China, Japan, Korea in the right, upper right, and the Philippines are just kind of in the middle on the right there a bit. And so this model will give you the technically the output is disease severity on a daily time step, which we turn into area under disease progress curve here. So you can see the dark areas are where the disease severity was the greatest and where it's lightest was the least. This particular model, also written by Serge, simulates unmanaged epidemics of bacterial leaf blight, brown spot, leaf blast, sheath blight, and tongue grow. Data requirements, precipitation and temperature, relative humidity, and a planting date just to start the model running. And the output is unmanaged disease severity, which we turn into AUDPC. So this is what the output looks like from EpiRice itself. We ran it using a time slice at 2000 for historical normals. And then we had 2030 and 2050 time slices, which basically means we had from 2020 to 2040 average climate data and 2040 to 2060 average climate data, which gives you 2030 and 2050 in the middle of those time slices. And you can see the disease progress curves basically are the same. This is the disease severity of the bacterial blight covering the leaf area for the historic 2000. And then in the future scenarios, we had 3 different emission scenarios, the A1B, the A2, and the B1, which are now the old scenarios, and we actually use different climate emission scenarios. When we look at the output from rice pest, if you're not familiar with violin plots, they're basically a mirrored histogram. So where the plot is the thickest is where you have the greatest amount of values, observed values. And so on the far left is the base 2000 historic data. And so what I want to emphasize here is you see that bulge just moves up a bit, which means that we start losing more yield in the future scenarios. We did all of this by taking the production situation, which had the fertilizer, the irrigation, and whatnot, and we used satellite-derived and georeferenced data. So we had the precipitation and temperature and radiation, and we used the same precipitation and temperature data in the 2 models, and we predicted the disease severity for rice blast and bacterial blight. And we used that disease severity data then to feed into the rice pest model and predict how much yield we were going to possibly lose. Excuse me. I'm only going to show you bacterial blight because, interestingly, when we ran the model, leaf blast became a non-issue. We didn't see any yield losses from leaf blast, and in fact, temperatures moved out of the favorable range enough for leaf blast that you saw improvements in yield, which is what you see here. If you see the blue parts of the map show where you're actually gaining yield, which means that you're not losing as much to bacterial leaf blight. And the areas that are red are those areas where you're starting to lose. So across the top is the 2030 time slice, and across the bottom is 2050. And then you have the 3 climate scenarios to compare. And there's a little bit of difference when you look at this, but for the most part, the thing that I thought was interesting was it mostly stays 0, but when you look at those graphs, you see that you actually do have, more loss. So those areas where you have more loss, you're losing quite a bit more, and in some cases up to almost 800 kilograms per hectare. So further work looking at guiding some breeding efforts. We were interested in looking at where disease and submergence or drought actually co-occurred. So this was part of Stress Tolerant Rice for Africa and South Asia, or STRASA, which was a Bill and Melinda Gates funded foundation project. And so this was a whole bunch of partners that were involved from Africa Rice and India, Bangladesh, and Nepal. And getting back to my GIS roots, I used the FAO's global administrative unit layers to delineate the just the administrative boundaries on the maps, and you'll see why this is important in a moment. We used Global Surface Summary of the Day, which is a product from the US National Center for Environmental, I can't remember what the I is now. It used to be the NCDC, the National Climatic Data Center, and then they changed it sometime, and I've forgotten what it stands for. We interpolated that station data into a gridded temperature layer, and we used Tropical Rainfall Measuring Mission, or TRMM data from satellite for the rainfall. So rainfall gets really tricky. Temperature is a relatively consistent value that you can interpolate over a surface, but rainfall is rather precarious. So we cut our losses because we could come up with a better temperature value but the rainfall we couldn't. So we took what we had available for rainfall and we put the two together. And I ran epi-rice model. And this is what epi-rice for bacterial blight output looks like with gall overlaid for India and Bangladesh and Nepal. And so this was just the AUDPC, which for plant pathologists means something, but I was meeting with plant pathologists to discuss this work, but I thought it might be easier if I presented it a bit differently. So I converted it into just the relative risk of bacterial blight. And in this case, I was in New Delhi, so we were just only interested in India. And I binned the categories by low, moderate, moderately hot, severe, and severe, and did this for bacterial blight and for brown spot. So in May of 2015, I went to New Delhi and met with partners. I showed them this map and said, what do you think about this? Is this, something that you believe? Is this useful? I want your expert opinion on this. And they looked at it and they scribbled on it and they came back to me and said, well, we think it really should look something more like this. So I took their notes. I went back to Los Baños. I modified my actual output files with another column to come up with this map, and then I recirculated it, and they gave me their feedback and said, yeah, that looks right. For brown spot, it was interesting because according to them, it severely underestimated the risk of brown spot when we did this. And so I want to highlight with this was an example of using a model where I knew it was wrong, but I went into it and said, look, I know this is wrong, but this is a way to start the conversation instead of just going into the room with a bunch of experts and asking them, what do you think, if I were to ask you to rank your area relative for brown spot or, leaf blast or bacterial blight, where do you think it ranks relative to the rest of India? That would have been much more difficult for them to quantify and taken much more time than it took me 15 or 20 minutes of conversation for each disease to have them draw on the maps and make some notes and give me some good hints that I was able to incorporate these changes. This is actually some work I'm doing now here in Deeperd in Western Australia. So I'm working with a group from Griffith University and from a colleague from University of Southern Queensland, still in the Center for Crop Health, looking at pathogen population monitoring. So I tend to do most of my work in R. And so this is actually a Shiny dashboard. So Shiny is an R package that allows you to make interactive web pages and dashboards and whatnot. And this was written by Ido Barr, who's a research fellow at Griffith University. And Ido and I have been working on this quite a bit lately, trying to get this polished up. So what we have here is Ascochyta rabii, which is the causal agent of Ascochyta blight and chickpea. Chickpea in Australia is a fairly profitable crop when you can grow it, but Ascochyta is always an issue. And so we're interested in the emergence of pathogenicity. And so on the right side here, you see this Agro ecological zone summary, and on the left, you've got the main dashboard. And if we actually just load up the layers, we can look at the agro eco zones. And so what we see here on the right is the zones, and corresponding to those zones is how many isolates have been collected from those zones and whether they've been genotyped and phenotyped and whatnot. And if you click on it, which I'm not going to do because this is just an image, but if you click on one of those numbers, then it zooms in and bursts out and you can drill down. We've de-identified all of the isolates so that they only go to the nearest town. And it's actually labeled 5 kilometers north of town because we don't want any farmers to actually be identifiable in this. But Ido's done a really nice job with setting this up. And I have been working with him to get a lot of the GIS stuff, like the layer of these agro eco zones you see on the map shown here, which is how the Grains Research and Development Corporation, or GRDC here in Australia, classify the growing areas of the grains regions of Australia. So GRDC, Grains Research Development Corporation, is a partially federally funded and levied organization that funds research and does outreach and whatnot. So a lot of our funding here in Australia comes from GRDC to do research in the grains area. And just as a side note, for those of you that probably may not be familiar with some of the funding structures and whatnot here in Australia. If you scroll down the page, we've got a graph that just shows the frequency of highly aggressive isolates. So the team in Griffith do the phenotyping and look at the aggressiveness on a set standards of chickpea cultivars, which you can see in the right side there. And then the yearly pathogen group frequencies, we can see right now that map only shows for 2020, but we've got quite a few years of data. And pathogen group 5, if I remember right, 4 and 5 are the ones that are starting to get more aggressive. And if you look back at the left at the line graph, you see that we're starting to see more aggressive pathogens emerging. For this project, this little bit, this shiny app is just a little bit of the project. I'm going to be developing, or more likely overseeing, the development of a model that we developed to help predict the emergence of aggressiveness in the pathogen population. Unfortunately, we've had a few hiccups in some of the data we're collecting, so I'm in the middle of changing my plans about what that model is going to look like and how I'm going to be doing that. But I'm suitably excited to be working on this, and I think it's going to be a really cool thing to do and see it all come together, and how we can actually tie that model in with this dashboard and show users, some benefit of a model. I'm going to switch gears a little bit now. So far I've been talking about instructing breeding. Now I'm going to actually talk about a bit of work I did looking at what happens after you've done the breeding and you've released varieties. So this was part of work I did at Erie for the Standing Panel on Impact Assessment, where we were trying to determine, was Erie still relevant, basically? Were the varietal releases that Erie was putting out showing any benefit? And so what we had was the average adoption of bacterial leaf blight resistant rice varieties in Indonesia and Philippines from 2001 to 2010. And we worked with government documents and surveys and expert opinion to figure out the Erie releases that went into the national programs that then released rice varieties in Indonesia and the Philippines. We then worked with experts to figure out when exactly did the pathogen overcome that resistance. So in some cases, something may have been released in 2001 and it was completely resistant to bacterial blight, and by 2010, the resistance was gone. And so when we were evaluating how much yield was saved, we looked at over time, starting here in 2001, it was completely resistant, so you had 100% yield, and by 2005, it started showing some chinks, and by 2007, it was completely resistant. So we rated it differently. And not only did we rate the varieties differently, but every province that you see on the map here, we had estimates from the experts on how much of the varieties was planted in those provinces. And so we were weighting the outputs of the models based on varietal adoption and resistance breakdown and whatnot. And so what we see is the Philippines had a much higher adoption rate of these resistant varieties for bacterial blight than Indonesia. But Sulawesi didn't do too bad here. But really the darkest, you had this huge percent adoption of 80 to 100% in most of the Philippines, which is really encouraging to see. And when we look at the effect or the tons per hectare, this is how much you were actually saving. Again, I did the epi rice and linked it with rice pest and came up with predictions of how much rice were we losing or how much were we actually saving if we had a resistant variety. And if you paid attention earlier, I said epi rice predicts susceptible varieties. And for this work, I just went through and said, well, we're just going to make an assumption that a resistant variety is 90% effective. And so I ran the model and decreased any losses by 90%, or the severity by 90%. And then we went back and we recalculated then everything else in between that 90 until the variety lost all of its resistance. And what we see, the Philippines, not surprisingly with the heavier adoption, the Philippines saw between 450 and 600 kilograms per hectare benefit from adopting bacterial blight resistant varieties. We didn't really go into why that was. We just wanted to see, was there an impact? It could have been proximity, because Erie is located right about in the middle there under my cursor, under that little white spot, which is Laguna de Bay, which is a body of water just east of Manila, and Erie is just to the south. We worked closely with Phil Rice. That's not to say we didn't work closely with the Indonesian government and their rice breeding programs as well. But when you're in the same country and it's just a day trip away, that probably makes a difference. It could be market differences. The rice preferences are an extremely interesting thing to see. The different countries and different regions have their very particular preferences for this type of rice or that type of rice. And Indonesia has been growing a variety of rice for many years that's very closely related to one of Erie's very first releases, IR64, called, in Indonesia's variety was Ceherong, because the consumers like it. So those sorts of things happen. Or it could be, as a plant pathologist, well, is there a difference in pathogen races? And, maybe there's something different about the Philippines than Indonesia. And so farmers in the Philippines saw more benefit from it. So they adopted it more, and therefore they saw greater benefits from adopting resistant varieties. Looking at developing new methods to address gaps in data availability. I've done quite a bit with climate change. So I showed you the climate change in Tanzania. This is going way back to when I was at Kansas State. I was working with Dr. Karen Garrett, who's now in Florida, University of Florida. And we were talking about, well, there's always stuff to be modeled. However, getting the data in the right format can be a real difficulty. And so we also have these policy needs where, again, this is an example of SIP wanted to know, where do we need to be thinking about climate change and late blight and the effects, and evaluating how is climate change going to affect late blight. And so we sat down and took the SimCast model, which was written by Bill Fry, and I used a version that Nick Grunwald had developed for his PhD work that incorporated resistance, highly resistant varieties into it, and developed a model written in R that used hourly weather data of SimCast. It only predicted blight units because we were only concerned with the biological portion. So SimCast has both a biological portion, which is driven by the weather conditions, but also a portion for fungicides like chlorothalonil that wash off. And so we, account for, you put on a fungicide and it washes off, because this model was written back in the 70s originally. And so I left that portion out, and I ran this model using hourly weather data and created a whole bunch of blight units. And I used that then to create what we called meta models, or models of the model, that used daily or monthly data that also predicted blight units. And to do this, I used weather data from the US. It was a fantastic weather data set that had hourly weather data for about 5 or 6 years. And you can see basically it covers all the continental US, a good bit of Alaska, some of Hawaii, and there's a little dot out in the Pacific, which is Guam. And when we checked, it represented most of the Kopp and Geiger climate schemes where you actually grow crops, which is good because potatoes are grown pretty much everywhere. So we called that good and said, yeah, right, we can do something with this globally. I promise you, this is the only equation in this talk on modeling. I try to keep them pretty light. I mean, it's late here and I know it's early for all of you in West Lafayette. Basically, blight units were the smoothed interaction of temperature and relative humidity, and K is a smoothing term. So if you've never used a generalized additive model, they're a really nice way where you think you have a relationship that may not be linear, and you want to account for that curvature. And so this figure that you see here on the x-axis, we'll call it, is the relative humidity. And on the y-axis, we have temperature. And on the z or z-axis, we have the blight units that are predicted. So as relative humidity increases, going from a little bit below 20 up to 100, and temperature, we went from minus 25 up to a little over 30. As they increase, the blight units themselves then increase. And so this was what we were trying to capture. And this is what that looks like. These are just box plots where the dotted line is a 1-to-1 and the blue line is fitted. And so across the x-axis are the blight units predicted by the original SimCast model. And on the y-axis are the blight units that were predicted by my generalized additive model. So we had pretty good agreement. You can see as it gets out here towards the higher values, it starts to drop off, but that was probably okay. We didn't worry about that too much because we thought, well, it's probably better, a little more conservative than not quite conservative enough. So we took that model, and I have a graduate certificate in GIS, which I was doing at the same time as my PhD here, and ran the model through a geographic information system with weather data and looked at different areas of the world. We singled out agro-eco regions that we thought were important. And in this case, we looked at Ecuador and Colombia for the Andes Mountains for potato production there, but also because of the origin of potatoes in the Andes. And we were curious about, is there going to be an issue with, late blight going into the future affecting wild relatives? And so you see this orange through here, that's basically an increase of 1 blight unit per growing season. And this darker orange is 2 blight units per growing season as compared to the base 1975 time slice. And so that was looking at 2050 A2. So this was the worst case climate scenario, which is still the trajectory that we're unfortunately on. So yes, it does appear to be an issue. Interestingly, though, around the time when I was doing this work, people tended to think that climate change was just going to make plant disease worse all over the place. And in a lot of times when I ran the model and I singled out a country and I looked at it, it was a lot like Tanzania, where we saw leaf blast became a non-issue. In this case, potato late blight became an issue in some places, especially these montane areas. In other areas where potato was grown, it was less of an issue. And as always with anything, it's a model. It's probably wrong, but it's instructive because we were running on a monthly time step, and we had to say, right, well, farmers plant in this month now. And so we're going, well, 50 years in the future, we're going to assume they plant at the same time, or we could assume that they'll plant when they'll get the optimal yield. So we shifted it to say, when do farmers get the optimal yield? By running a very simple crop growth model using the same climate data that I use to inform this model to predict these sorts of things. And in the paddock, in the moment, tactical decision-making, this is, well, yeah, I've been in Australia long enough. I've started referring to the field as the paddock. This is a project that I'm working on here in Perth. It's a pretty cool project. I had the good opportunity when I joined to take over the maintenance of a model. So one of my colleagues, Beck O'Leary in Perth, had already started rewriting a model that was written in Excel that predicts when canola blackleg spores or Leptosphaeria maculans spores are going to mature, and whether or not you needed to spray fungicide on your canola crop. And previously, we had to load up Excel and put a bunch of weather data in, and then look at the Excel spreadsheet and send this stuff off. And we've gotten to the point now where I can receive a request. I can go generate an output. This is an example of a Word document output that's generated. Badgingarra is a weather station here in Western Australia. Farmers then can go to our website after this has all been approved by experts and look and see if they're in Badgingarra and they sowed on April 29th of 2021, they're at moderate risk of exposure. But if they sowed on 13 May of 2021, they were at high risk of exposure. So we're trying to automate a lot of these things. This is going to be further automated through the use of APIs or application program interfaces, where we have this spore maturity risk that feeds into an app. So what you're looking at here is a screenshot from an iPad app that we developed then, that doesn't tell you what your spore maturity risk is, but it's kind of a two-step thing. You can then go use this app and feed in your situation, and where it says LMH, that's where you can put in the values from the previous slide. And this app will then help you determine, is a fungicide going to be effective? Am I going to lose money? Am I going to make money? You know, is it going to be beneficial for me or not? We have a whole suite of these sorts of apps. We've got one that we just released for tan spot and wheat this year, but don't look for it called tan spot. It's called Yellow Spot WM because we like to do things differently here in Australia. We have one on powdery mildew for mung bean, which I worked to develop quite a bit while I was still back in Queensland. We have a stripe rust for wheat, and we have a sclerotinia for canola, and we're working on a couple others right now as well for an upper canopy infection for blackleg in canola and whatnot. So we've got a really good team of people doing a lot of work on developing these apps for iPads. We've started releasing them on iPhone as well. So the wheat yellow spot or tan spot app is actually available on iPhones now as well. Some other work that you might think about if you're in the paddock and you're trying to make a decision. This is looking at field pea black spot spore showers. So this model exists. Previously, it was a wind dispersal model, and it was like, how far does the wind disperse the spores, but it wasn't really spatially enabled. And so this was work that CSIRO did here in Australia to identify crops using satellite imagery. In this case, it was Sentinel-2. And the green areas show where field pea was growing when the satellite passed overhead. And then we've started using this to identify last season, this paddock, this field was field pea. And then we run our model. And when we run our model, this is what it looks like. So on the left side is the nearest weather station is in the bottom right corner, and on the right side, the nearest weather station in the upper left-hand corner. And for a rain event where there was some wind, you can see that the wind came through and spread spores around. So the idea is that ultimately we want to get to the point where farmers or agronomists can start looking at these maps. And with this particular disease, you know that after a while, the spore showers are going to stop, the ascospores are going to be spent, and you can safely sow your field pea crop. And so this helps you decide, well, you know, if the wind is reliably out of the south and my field is down here to the south and it's not in the prevailing direction, then I probably don't have as much to worry about because I didn't get as many spores landing in my crop. Or maybe you're just really risk averse and you're looking at going, well, that's still too close. I'm still going to wait until I don't see any more spores that are being ejected and landing anywhere nearby. This goes along with my bioeconomic modeling hat now. Now that I'm no longer officially just a plant pathologist, I'm developing a whole farm liming model that uses Monte Carlo simulations. So we have a liming model that does a single paddock at a time. And I'm working to develop a framework that is capable of seamlessly ingesting data from farmers so that they don't really have to do too much work on their end, run their entire farm through a Monte Carlo simulation using weather data, and predict many future possible outcomes. So what we see here is the likely gain over a 20-year period. So where the graph is the thickest, again, is your most likely value. The mean is 93, which is not your most likely value. Your most likely value is probably closer to 75. So you're probably going to earn $75 eventually by following this liming strategy versus not doing anything at all. But you have this little chance out here of losing up to $50 per hectare. And this risk side shows how big is your debt going to get before you start earning back. And in this case, you're likely to lose $53 per hectare as you've put on lime before you start turning things around, because we know liming isn't one of those things like fertilizer. It takes lime a while to start paying off. The idea behind this model is we've already got the iLime model, and I can do the framework in R or Julia for the Monte Carlo simulations and show farmers the benefit of this. But also we want to build something that we can expand into, should I buy that property next door? Is that going to pay off? Or do I hire someone else to, help work on the farm? Or do I buy that new tractor? Or should we build that new shed? These sorts of big investments that look at cash flow over time are the types of things that I'm going to start looking at in this model framework once I get it up and going. So I just want to reiterate, essentially all models are wrong, but some are useful. And I think I've certainly found good use in all of these that I've shown you. Hopefully they've been useful to the people that I generated them for as well. And hopefully you've gotten some entertainment and use out of seeing them. I'm going to switch gears quite a bit now and actually talk about open and reproducible research and open plant pathology, just because this is something near and dear to my heart. And I was one of the things that was specifically asked for when I was invited to give my presentation here this evening. So if we think about an ideal simplified research lifestyle, you collect your data, you analyze it, you write it up, and you publish it. And if we think about this then as adding a layer of openness and reproducibility on top, when you collect the data, then you can share it in an open repository. When you analyze it, you can share your code. If you use R, Julia, Python, you can share that code. Anybody can use it. I have to say this really hit home when I started working at Erie, and I'm working with country partners that didn't have a budget to buy SaaS or those sorts of things. And, they could just take my code, my R code, and go use it. But even if you use SaaS or something like that, there's still benefit in sharing the code because other people can pick it up and learn from it and use it. And when we write, use collaborative tools like Google Docs. Word now actually in Teams is actually not too bad most of the time. And when you publish things, I know open access is not cheap. And if you don't have that in the budget, a preprint goes a long way. That's a good way to get your science out there. And especially this goes back to, like I said, a lot of these developing countries don't have these big budgets. Preprints are a good way for them to get access to the data. Why do we want to embrace open practices? Well, in some cases, sponsors and journals are starting to require the data to be more open. This is becoming the standard in molecular data where you have to put it in the database, but also we're starting to see donors that are requiring it. The Gates Foundation starting to require it. Federally funded research here in Australia requires it. There's other things that I can't think of off the top of my head where I know it's necessary. Allows reproducibility for data and or methods. And oftentimes I think we tend to think this is for other people, but I can't tell you how many times this has been useful to me before anyone else. because, I get those comments back on that manuscript and I have to go back and I have to recreate a figure, or I have to figure out what I did, or somebody comes to me and says, hey, I had a question. You did this in that model and I'm trying to do something. And I can say, well, yeah, right. Here's what I did, and, have a go at this. The technology is becoming less cumbersome and more available. We now have things like GitHub, which we didn't have 10 or 15 years ago, and makes sharing your code so much easier, and finding other people that are sharing similar code very easy. Things like RStudio and that company are developing tools that make it much easier to make reproducible research, things like R markdown files and whatnot. We want to embrace it because it gives you enhanced visibility and transparency. We know there's a reproducibility crisis in science. This is a good way to combat that. It also raises the visibility of your research because you have more things out there, more ways for people to see it, which leads to multiple citable outcomes. If you do it right and you deposit your data in a good data repository, not code repository, a data repository like Zenodo or OSF or Figshare, you can mint a DOI, and then people can cite your data. The code, same way, you can put it up and have it cited. And of course, you get the manuscript out of it as well. What are some barriers? Often there's a lack of interest, or maybe people just aren't aware that you can do this. We really don't see a lot of supplemental information posted. There's low incentive and there's not much pressure, but that may change. We're starting to see more and more that there's requirements to post the data. There is the idea that it takes huge time and effort. And I think that's also I'd counter that and say, I think that it is like anything, it's a learning curve, but this goes back to that reproducibility thing. In the long run, it saves me so much time because everything is well organized. And I have code from my PhD and my first postdoc that I can still go back to and still run. It's still reproducible. Documenting the data and the code really helps when you start writing your manuscripts. Versioning code and maintaining those versions. This goes a little bit deeper, but you start using version control like Git and whatnot. People get scared when they see that. It's understandable. But once you learn it, then you probably want to start using it with pretty much everything. And of course, I'm not going to deny there is the fear of being scooped, but I'm going to counter with saying I've had a lot of things that I've had posted on GitHub as I've been working on them, in the manuscripts and whatnot I've been writing. And maybe people just don't really think that what I'm doing is worthwhile, but nobody's ever really scooped my stuff. But also, if you do it right, you've got the preprint out there and you've got the DOIs and whatnot, you've already got your flag out there and you've established that you're doing this. So with these sorts of things in mind, seeing all these barriers and all these problems, Emerson Del Ponte, who's in Brazil, and I decided that we wanted to change that around 2018. And so we started Open Plant Pathology and set up a web page at openplantpathology.org. And we've joked that, we're both in the Southern Hemisphere, but we're on opposite sides of the globe. So we have 24/7 service that one of us is usually going to be awake if you need any reproducibility questions addressed. So Open Plant Pathology's vision and mission is pretty simple. We want to foster a diverse community culture that values open, transparent, reproducible research using shared data and reusable software. And so we did that. We set up a Slack. It's not a super high traffic, but I have to say it's a very collegial place. And there's a lot of really nice folks there. So feel free to go visit our website. You can sign up on our website. You don't need an invite. It's a good way to expand your network. If you're a graduate student, it's a good way to get involved and meet some of the experts in the field that you may not be exposed to, especially right now with COVID where you can't travel and whatnot. But we have a little over 300 people, I think, that are members all over the world. When we went beyond just saying we're going to set up a Slack, we have Twitter. where we try to tweet things related to open science and promote open science reproducible research. We have GitHub, where we store code and data files. We maintain a data repository on OSF. We have a website and a blog. And I noticed that we don't have our YouTube listed in there, which is our next slide. We have a YouTube channel, which has over 200 subscribers. I've been putting up and archiving videos from the Australasian Plant Pathology Society Queensland chapter recently. Where seminars like this are recorded, and we're happy to put that up and host an abstract on our website. We mint a DOI for the presentation video and everything, and make it all available. We also have a series of seminars that we did that were just open plant pathology seminars that we did last year when COVID struck. So there's a series of a few seminars that are not related with the APPS or APS or any of those sorts of societies. This is just an idea of our Twitter when we were just trying to get things up and going. We've got our GitHub, where this is our central hub of where we do most things related to development of the website and related to some R packages that we maintain. Our website has since been redesigned, but we have blog posts where we may do something. I've got one where I do linear regression, show how to do linear regression of modeling nematode populations in the soil with some work from John Thompson, who's a retired professor at University of Southern Queensland Center for Crop Health. And I've made it all reproducible and show you how to go through and do linear regression in R and show you how to evaluate the fit of the model and why you'd want to and all these sorts of things. We've done a series of interviews with people like Sofian Kamoun and Ben Marwick and Jade Diplom. So Sofian, we talked about open research and open tools to advance the field. Ben Marwick and Jade Diplom, you probably may not know, but they're archaeologists who are very fond of open science and open practices. And so we asked them to come along and give their thoughts on why they use open science and open research. I mentioned we have an R package. Hi, Austin, I saw you signed in. Austin McCoy, who's also a Boilermaker, now at Michigan State, approached me a while back and had an idea from his PhD research that we've turned into an R package that's published in NPMI and has a couple citations on it. This was a really cool project. I'm really glad that Austin took the initiative to come to me. So he came up with the idea, and I have the experience writing the R packages. So I was happy to write the R package and maintain it on CRAN for him. So this is entirely an open plant pathology initiative here for this R package. And if you do any population work on population diversities, especially with Phytophthora soji, you probably know this one. Previously, we've done quite a few workshops. Last year, COVID slowed us down quite a bit, but we've done network analysis in plant pathology and introduction to multivariate statistics using R and population genomics. Here in Australia, I did one for R Markdown for scientists. And currently we don't have any workshops planned yet, but I was seeing that APPS has the call out for workshops for the next APPS meeting. So I may have to put my hand up and do another one. And wrapping things up, just eating my own dog food for breakfast here. We've got repositories for Africa rice, Tanzania work, Strasa, and the meta modeling, late blight, and for the R package resources. So here's the links to some R packages. You could recreate a lot of this using EpiCrop, which does epi rice. This one's in heavy development right now. That one does the disease severity, the GSOD-R, NASA Power, our daily weather data. Chirps is another one that does rainfall and temperature now. This was written by Kai de Sousa. I'm a co-author on that package. Get Crew CL 2.0 is climate data. And of course, Haggis, Austin's package is available. So if anybody wants the slides, I will make these available online later. And of course, all the links will be available as well. And with that, say thanks, and I'll stop sharing my screen if I can figure out how to do that and take questions.
Are there any questions? And I want to encourage the graduate students and postdocs to go ahead and chime in. Well, I can start real quick because I do have a quick question. I'm old enough to remember when the internet was going to be a happy place. And of course,
Yes.
here we are. So I'm wondering, when you're designing a sort of transparent system like this, or an open access system, do you go in thinking about how the system might be gamed or misused and sort of head that off at the pass rather than, and I don't know what that would be for this, or if there would be any potential problems?
Look, well, that's the same sort of thing with most of our research or with our models. You know, you can misuse it, and you have no control over how it's misused per se. So I typically just know that it could happen and just don't worry about it too much because it hasn't happened yet.
That's the spirit.
I'm not sure how you'd actually do that if you're actually going to be open, you know.
Sure.
It's going to be really difficult. And I mean, I know a lot of other scientists that don't want other people using their models because, well, somebody might misuse it. Well, yeah, they might misuse it, but in a lot of cases, I've had a lot of people contact me and say, hey, I found this code on GitHub for this EpiCrop package you're working on. And, you know, and there's been a student recently from West Africa who contacted me and said, I'm trying to run it and I'm having this problem. So I helped him through it. And he's like, oh my gosh, that's so great. You know, I've got this now for my PhD research. The benefits far outseed the negatives.
Gotcha. Yeah, I tend to agree. I used to be skeptical, but you just watch it in action. It's like that. It's so impressive.
Yeah.
Any questions from the audience?
I do have one, but let's see if a student or a postdoc has a question.
Come on, guys, don't be shy.
I have one. So
Yes, please.
do you have, well, thank you so much for your presentation. And my question is related to the open plant pathology. So do you have a specific like protocols like for sharing documents or the data there in your website?
We don't have specific protocols, no, but open plant pathology is the kind of place that if you have questions, if you say, I want to share this stuff, it'd be like Austin coming to us and saying, I've got this R code and I want to do something with it. We have people that can say, yeah, right, you have that sort of data. Here's the type of thing you need to think about and how you can share it. So we don't necessarily have specific protocols, but we have the people that know and can help you through that process.
Okay, thank you.
So here my question as we wait for others. So you were talking about one of your projects on climate change and how you and your team had identified that the impact will not be equal, you know, locations, which is fantastic. One example, you gave the example in Ecuador where you found the increase in blight units, actually 2 per season. So that's very informative. Now, what is your actual suggestion in terms of management, actually in the future? So what tools do you think we need right now that have not been implemented yet in order to identify those areas at risk and improve things? Because it seems to me that in most cases, we treat everything equally in terms of pesticides, in terms of deployment of genes, everything, but that might be the case. So what should
I've got a paper on that one too, about the deployment of genes. Yeah, and that was one of the things that we, with the late blight stuff, that was one of the things we really tried to highlight was this isn't equal. I mean, yeah, in Ecuador, we took and even then it's still a fairly broad look. The patterns that came out it was the montane areas. And so I think that was where modeling was useful. If you wanted to say, what can we do right now? We have the varieties that have been developed using wild relatives to bring resistance genes in. We can use those, but then that goes then on to, we put that absolutely everywhere, which you alluded to, Christian. And we've, in a perfect world, if I get up on my soapbox here, in a perfect world, if we think about deployment of genes, if we monitor pathogen populations in something like bacterial blight and rice, where we know there's a strong gene for gene interaction, and we know which genes are in this rice variety and which genes are in this rice variety, and we know that that population of Xanthomonas in this particular area has these genes, then we can deploy the proper rice variety there and start rotating those things in and out. That's in a perfect world. You have to really work with country level governments and seed companies and whatnot. Ideally, that would work. I don't know if that would actually work, but it would be really cool to see something like that. You know, that's the type of thing we need to be thinking about doing, rather than just the blanket, well, we've got this, it works, we use it everywhere. That's what gets us back to, well, now we have to figure out what to do because the pathogen, not surprisingly, overcame the resistance here.
Very good, thanks. Anybody else? Questions?
I have a question. So could this model also be used for predicting new introductions of disease? Let's say there's a disease that we haven't really been watching as closely as maybe we should, just because right now the climate in a certain area isn't very conducive to it. This model help us be more proactive against new introductions of disease into climates that they might take off in.
I'm trying to think. You could use some of the models I showed you in that fashion, but I'm aware that actually in the US there is a model framework that was written that I think APHIS uses. It's just a generic framework that's that sort of thing that you put in basically the generic temperatures and your values for upper and lower minimums and what the relative humidity, whatnot. If you've got what you think is a new pathogen coming in, and you can just run this model and see where it's likely to become established. There's also lots of models that are ecological niche models that are a little bit more advanced that you could do that sort of thing with and start using climate data in, like Climex and GARP and some of those models. Ultimately, the models I showed you, I think the epi-rice model is simple enough. It has simple enough outputs. I didn't show the original paper because I didn't write the original paper. I've just used the model quite a bit, but in the original paper, it shows that tongue grow could be a problem in Africa where it doesn't actually occur in Africa anywhere. So it's two viruses that don't, and the vector don't occur in Africa. So you could look at it from that perspective and say, well, yeah, we could start looking at some climate things and running climate scenarios and through this model and see where does it start increasing, just like I saw with leaf blast, the data that I didn't show you because there was no data to show that it became a non-issue. Long-winded answer to say, yes, those things are all possible.
Thank you.
Okay, so I think that if we don't have more questions, we're gonna have a break, and we will connect at 10:15.
Cool.
We are gonna have like the conversation with the grad students and postdocs.
Okay,
Sounds
Amy, can you make Mariella the host so you don't have to hang out?
Yes, Mariella and Becca, both are
Excellent.
the alternate hosts for the next meeting. So that should be in your meeting invite, everybody. Thank you, Dr. Sparks.
Thank
No
you so
worries.
much. I'm going to go out and harvest. You've inspired me.
Thanks, Adam.
Bye-bye.
Good to see you.